genomes and transcriptomes with species with what I'm calling complex genomes, and I'll explain that in a little bit more detail. But just to kind of give you a little bit of a roadmap where we're going to, a little bit about my research interests and backgrounds, and I'm talking about 3 different projects that are collaborative efforts. One is with flower size in Nicotiana, and that's in collaboration with Elizabeth McCarthy, who's an assistant professor at SUNY Cortland. And then we'll move on to floral variation in Calochortus venistus, and that's collaboration with Adriana Hernandez, who's a PhD student here in Chelsea Specs Lab at Cornell. And then lastly, Larea, or the creosote bush, polyploid evolution, and that's in collaboration with Rob Lepore at Rhodes College. I do want to point out that all 3 of these projects are not NSF funded, so I'm going to show some things that you can do on a kind of a smaller budget for PhD students and postdocs. My research interests are broadly encompassed with adaptation. So here's some classic examples of adaptation. We have Darwin's finches, and if I can figure out how to make this work. So we have Darwin's finches here that we hear about in intro bio. We have the cichlids in Africa, peppered moths, and then also some examples in plants, and those are some of the examples I'll be looking at. Current types of projects do span a wide range. They are at the population level. Example in Solanum habrochytes, looking at levels of heterozygosity and population structure. 2 angiosperm-wide studies, such as whole genome duplication, which kind of Fei talked about just a little bit. So whole genome duplication across the angiosperms. And then so in my title, I talk about complex genomes. And so I'm going to focus on genome size and polyploidy for this talk. So some examples. So here are data for the distribution of genome sizes in the angiosperms. Arabidopsis thaliana, the model of the plant world, is about the same size as some of Fei-Wei's hornworts. Easy to work on. Close to the average, or not the average, the mode, sorry, of angiosperm genome size is rice, around 400 or 500 megabases. The median value is close to ginger, around 1500, 1700 megabases. The mean is around barley of 5 gigabases. And then you get to some cases like onion, 16 gigabases, and tulip, over 59 gigabases. And this is genome size. So the bigger it is, the harder it is to sequence. One of the projects we're talking about today is Calochortus venistus. It has a genome size of 5.5 gigabases, so it is definitely bigger than the mean, but not as big as it could be. The other term, or the thing that makes genomes complex, is polyploidy. And so polyploidy is having more than 2 sets of chromosomes. So here's an example. We have parent 1 hybridizes with parent 2, and now instead of having 2 chromosomes, these long ones, now it has 4, 2 from each parent. Polyploidy home genome duplication was initially thought to be an evolutionary dead end, but more recent studies have suggested that having this more chromosome information, more genome information, may actually help plants survive or thrive in some cases, such as disturbed environments or climate change or herbivores. And polyploidy is also very common in a lot of crops. Wheat, cotton, strawberry, etc. And so in strawberry, you have the diploid fruit, which is much, much smaller than a polyploid fruit. A couple other things I want to talk about just real quick. So flower size, this would be involved for some of the projects. So flower size is very important for reproduction. So you have this Christmas star orchid, and it is pollinated by this hawk moth whose proboscis is long enough to reach into the nectar spur to get nectar. Nothing else can pollinate that. So there's a strong coevolution between the plants and their pollinators, and this facilitates more efficient pollinators while negating or excluding nectar robbers, pollen thieves, etc. And we see that cell size and cell number can both play a role in the flower size. So here's some work I've done previously measuring cell size along the corolla. And some examples in Aquilegia have looked at the similar things in the nectar spurs, so to show that cell size is very important for that. So these different types of flower sizes, you have different types of pollinators. So we have hummingbird flowers are often long, red, tubular flowers. Hawk moths are usually long as well, more narrow and white. And then things that are pollinated by bees are more open, bowl-shaped. And then you have butterfly pollinated flowers, which are really small, where the butterfly can land on. And so thinking about that, that moves into the first project of Nicotiana, looking at floral diversity. So here's the tree of the genus that was published this year, and with the flower sizes associated with those. So you can see throughout the genus, there's a very wide range of flower sizes, from very small things to really big ones, the Lotta, and then also Sylvestris. And here's tobacco right there, which is a polyploid. And so with the polyploid, we have two progenitors that have formed tobacco. Sylvestris is this long-flowered individual, which was the maternal parent. And the paternal parent was Tomatosiformis, which is much smaller. So we have natural hybrids that were formed about 0.6 million years ago that we can look at. And we also have been able to create synthetic polyploids in the lab using the same crosses. And you can see, even though they had the same, or pretty much the same genomic makeup, they do look quite different, both in terms of flower color, flower size, flower shape. And so we want to look at that and to see what's going on. There are, for these three species, there are reference genomes previously available when we started working on this project. And here's some nanoplot examples of those. So we have the two progenitors, and these were sequenced several years ago. We have about 250-some thousand contigs of the maternal parent and about 160,000 contigs of the paternal parent. The median contigs are not very big. The N50s are okay, and there's assembling not all of, but quite a bit of the genome. Then there have tobacco right here. It has over 1 million contigs and fairly larger N50 values, but it's missing more of the genome. So even though a lot of the genome is missing, if you look at the Busco genes for just things in the Solanaceae, you do see that these genomes do encompass most of those single-copied nuclear genes that you should have there. We do see, though, that the polyploids, even though they should all be duplicated, not all the genes are. So that does lead to some problems with trying to map back RNA-seq data. Now, we did try to use these genomes to do that, but we had very bad success with that. And so we are looking at RNA-seq for this study with the progenitors, natural, and synthetic polyploids. Again, with some of the shortcomings of the reference genomes, we did take a de novo approach. We have flower material, again, for essentially 6 different taxa, three different developmental time points through growth. We have the Illumina data for a lot of the differential gene expression, and we did include one flow cell of nanopore data. And the short reads were very difficult to assemble and distinguish between homologs, or if we have one gene determining if it came from the mom or from the dad. And so with the nanopore data, we originally tried to do cDNA direct sequencing, but we couldn't isolate 1 microgram of mRNA, which is what is recommended by the kit. So then we went on to doing the PCR amplified cDNA. We barcoded 4 different tissue types in one flow cell, and that gave us a little bit over 6 million reads. So we had 2 different species represented here, so about 3 million reads per species. And on the top here, you can see the distribution of the read lengths. So a lot of them are close to that 1 kb. This was using that cDNA kit, which uses that transposase enzyme that we were talking about this morning, and so it does shear some of the DNA. But we were getting quite a bit that was large. These smaller fragments here were wound up being excluded from further analyses. And then we did error correct this data with the Illumina data using FMLRC, and assembled it with both short and long reads with different assemblers. So I'm just going to compare a little bit here. So we use Trinity, Transabyss, and Spades. We have the proportion of Busco genes from Solanaceae with the different types of data and different assemblers. And so we do see quite a bit of change in Busco score. So if we look at just the Illumina data, the blue is complete genes. We see mostly blue, but we have about 400 and some missing Busco genes completely. If we use Trinity, which is a very common assembler for RNA-seq data, it actually performed the worst for us. We had a lot more missing data in that case. And then Transabyss and Spades had the fewest amount of missing data, the most complete single copy genes, and this is just for one of the 2 progenitors. So this is not from the polyploid. So we do see changes in BCBC genes and also changes in the number of contigs and the N50. Part of the difference I think we see in the number of contigs is how some of these programs deal with different isoforms or different splice sites. So you may have this transcript splice site here, and things can come together in different ways. At least for Trinity, using like the supertrans option, it makes one transcript and puts it together, so you can use that to map things to. So that's a little bit of the reason we have some different number of contigs in this assembly. But the basic, one of the take-home message I want to highlight here is that the hybrid approach does help with transcriptomics, especially if you already have Illumina short reads. And so adding in just 3 million long reads for each species, we were able to bring down the number of contigs by over 10% in our assembly, and we boosted the N50 values by at least 200 bases. By removing some of those really short nanopore data, so keeping only things that are 800 bases or more in the long reads, actually boosted both of those more. So we had longer N50s and fewer contigs overall. And so here's a comparison again between those two plots where we're getting rid of a lot of the smaller things to make sure things are assembling together. So now that we have these assemblies, we can ask some different questions. And so the two things that jump out at first are looking at flower size and flower color. I'm going to touch on a little bit of the results for flower size. If you think the flower color stuff is exciting, I'm not going to talk about today, but tomorrow at 1 p.m. Elizabeth will be talking about this, so she'll go in much more detail about the results that we found here. But with the mindset of flower size, so if you look at differential gene expression using the Illumina data, if you compare two progenitors, we see a ton of genes that are differentially expressed. So things that are red are differentially expressed genes, things that are black are not, which is not surprising given how different those flowers look. If you take the natural polyploids, they have a few differentially expressed genes, not near as many. And the flowers do look more similar, but they're quite different. So we can focus on some of these genes now that are differentially expressed. Even though these have the same genomic background, we can see maybe what's driving some of these differences that we see in flower size. And so I'm going to highlight two examples of this. So this plot is a lot going on. The more transparent data up on top, or we see some examples on top here, are from N. sylvestris, the maternal progenitor, the longer flower of the two. The darker circles are from the paternal smaller parent. And we go through different sizes of from stage 60 to 85 to 95% enthesis. And even though there's a lot of data here, one thing I would do highlight is that for these longer flowered individuals, we see that the genes from the maternal are upregulated. So the genes that are for big brother, in this case, one organ size gene, is more highly expressed in these longer flowers than the copy from the smaller parent. We see something similar along a folia. It's a little bit harder to see in some cases, so we do have the paternal parent, the short flowered one, having fairly high expression of some of these genes. So we need to look more into across the flower what is going on, but we are seeing differentially expressed genes in some of these known candidates for organ size. And so just a couple of conclusions from this part of the project. So adding the Nanopore data really did help us in improving the assembly of the transcriptome, which is probably not surprising. And then all assemblers are not created equal. I know Trinity is used a lot. I've used it a lot in the past. But at least for the hybrid option, it actually performs one of the worst. And we had problems running that actually with a lot of the data we had. It's just too much data. It couldn't handle it. The majority of the genes are differentially expressed between the progenitors, and then there's drastically fewer of those between polyploids, but now we can focus on what is actually driving some of these differences that we see. And some of these are in candidate genes for organ size. And so we want to look at that more closely. All right, so I want to switch a little bit to another project. So this is looking at lilies. So when you think of lilies, most people think of examples here, where you have the outer whorls, both the petals and the sepals are both very showy, very colorful. And these are the true lilies. But the true lilies are about 100 species and only make up about 100 out of 1,700 species in the order of the Liliales, which represent about 100 to 120 million years of evolution. If you look at all of the Liliales, the flowers are actually quite diverse. And so here's some examples. Tulip, also very common that we know. Smilax, the flowers are not as colorful, very reduced. And then you get to things like Calochortus over here. They're very showy, very different from your true lilies. And the Calochortus, within a species, we have to see a lot of variation as well. So Adriana is looking at Calochortus venistus. This is all from the same species. You can see the range of flower color. We got reds, whites, pinks, yellows. We have nectar guides, color spots. And so there's a lot going on within the species. And so she has spent the last 2 summers in the field in California where these things are found, collecting samples. And so this is a graph that she has produced. So this is California here, and all those blue dots are herbarium records where populations have known to have been found at one point. And she's gone through a lot of these and counted the number of different individuals with different colors. And so if you look at this population up here, most of this proportion, a large proportion of the populations are all white-flowered individuals. You have some that are yellow and some that are pink. When you move down to the southern tip, you see a lot more reds, purples, pinks, very few yellows, and then some more, and the rest are made up of white individuals. So there's a lot going on here with just the flower color. And so some of the research questions that she's interested in and I'm trying to help are there taxonomic groupings based on floral phenotypes? What does the population structure and genetic diversity of Calochortus venistus look like in California? And then can we answer the question of where did the species originate and how is it diversified? So we're trying to employ PopGen approach to try to answer these and other questions. And so to do that, it'd be helpful to have a reference genome. So Fei-Wei kind of talked about how overrepresented angiosperms are compared to other groups. So from a paper last year, there's at least 236 sequenced genomes out there, probably closer to 300, 400 now. It's hard to keep up. There's new ones all the time. But I do want to highlight that they're not spread across the angiosperms, the flowering plants, equally. A lot of them are the eudicots. There's more species there, but there's also a lot more reference to genomes. The liliales are in the monocots down here, and so there's very few in the monocots, but how do they compare phylogenetically to Calochortus? So taking the angiosperm tree of life and parsing it down to 1 tip per family, Calochortus is this blue box here in Liliaceae. These red boxes represent the other known Liliales genomes, at least from that 2018 paper, and there's nothing close. The closest one is in Asparagales, which represents over 123 million years of evolution. So it is not a very good reference to use for PopGen analyses. And so one other thing I want to talk about before we get into what we've been doing with trying to assemble these large genomes, so again 5.5 gigabases, there's many issues that are very well known. Many assemblers that are out there are designed for genomes the same size or smaller to the human genome, so 3 gigs or less. And then the large genomes themselves are more repetitive, but have fewer, are about the same number of genes as smaller genomes. And we've already heard this morning and this afternoon how repetitive aspects are really hard for assembly, can kill Nanopore, Lumina, all those things. So it's very difficult. And you also have the computational resources for large genomes are pretty intense, both in terms of memory and wall time. So here's one example from the Abyss paper that came out, Abyss 2.0, where you have peak memory in gigabases, or gigabytes, sorry, on the y-axis, and wall clock in hours along the right. And if you look at big genomes, I think this one actually is used the human genome. You can see some of these programs may take over 1 terabyte of RAM and 1,000 hours, computationally speaking. And so there's a lot to go into that. And so for Calochortus, again, we're kind of on a tight budget here, so we generated 240 gigabases of Illumina short reads, it's about 40x coverage, and about 40 gigabases of long reads, about 8x coverage. So not super great, but it's a good start. We're also using the rapid sequencing kit. So again, you just take your DNA, it uses the transpoase to break it apart in some cases and ligate the adapters. And we try to remove the short read fragments by using the short read eliminator, but that was about this morning and Fei-Wei mentioned as well. So we ran one flow cell without the SRE. You can see that we have a really big peak on very small things. We did not optimize the DNA extraction in this case for long reads, but we weren't getting very good results here. We ran the short read eliminator kit and with bioanalyzer trace. So this is 6,000 bases right here. We got rid of basically everything below 6,000, and we see a nice little peak of our high molecular weight. Now we can't say how big this is because we didn't use the equipment that was mentioned this morning. But when we sequence this, now we're shifting that peak to about 4.5 to 6 kb. So not super long fragments, but we're getting a much better result now sequencing wise. And one thing I would mention, I did truncate these plots, but we were getting some reads that were over 500 kb. Not a whole lot, but we're getting some very long fragments. So for the sequencing and processing, this is the nitty-gritty. I can talk more about this later if we want to, but we use PoreChop to remove adapters, NanoFilter to remove very low quality things, error corrected again with FMLRC. We did use the Illumina data to assemble because that was where a lot of our data is, but we normalized to 50x coverage to try to remove some of the repetitiveness. And doing that actually helped a lot with our data requirements, our computational requirements. So to assemble an abyss with the normalized data required only 475 gigabytes of RAM. Doing that full data set took over 700 gigabytes of RAM and would take 4 days just to read in the data itself. Whereas with the normalized it took about 2 days. So it does help to remove that repetitiveness if you don't need that in assembly. So we did use Abyss to assemble using both Illumina and then Illumina and Nanopore. And for just the long reads, even though we didn't have much coverage, we used WTDBG2. We did try to use SPADES, which is very common in bacterial genomes to use, but it could not handle all the data that we had. So here's a comparison between the three of the different assemblies. Again, our total genome size is about 5.5 gigabases. So with just the long reads, again, we didn't have much, but we had an N50 of 14,000. We had 33,000 contigs, but we were only assembling about 400 megabases of the genome, so about the size of rice. So we're missing a ton of things here. You take just the Illumina data, we have now 35 million reads, or contigs, but we're assembling about 3 gigabases. So we're starting to move there. The N50s are very low. You use the Nanopore data to scaffold, so even though those didn't do very well in just the assembly by themselves, you add that to the scaffold. We do cut down our number of reads quite a bit. We still have a ton, but we are now assembling 4.3 out of the 5.5 gigabases. So we don't have a complete assembly for sure, but we're making progress. If you look at those Busco genes, compared to 91% that Feiwei showed, we're awful. However, we do see that the Nanopore only, we have 4 of the 1,000 or the 1,300 genes. So we don't have many at all. We're missing a ton. But with the Nanopore and Illumina, we're starting to bump up there a little bit. Most of it's fragmented. So we wouldn't want to publish this. This is not a great genome. However, it is useful for some things. So again, we want to do some PopGen for this main project. So using some preliminary RADseq data that we have in the lab, we called SNPs using stacks, both in a de novo framework and a reference-based framework, and did some filtering. And what we see along the bottom here is that if we use a de novo, we're only calling about 6,000 SNPs total. We start using these different assemblies, even though they're not great Busco-wise, but when we get up to the hybrid assembly that combines all our data, now we're calling SNPs on about 250,000 SNPs. So now we're starting to get into the realm of where we can actually do some good PopGen work, even though our genome is not great, we're getting some good results. And so Adriana spent time in the field again this summer, collected 192 samples that are being sequenced for RADseq right now. When she gets those back, we're going to use the hybrid assembly to map to that so she can start looking at PopGen questions. And so just a few conclusions from here. You don't need a great genome for making big improvements for PopGen work. We are assembling a decent amount of the genome, but we're still missing a lot. But normalizing the data helped a ton, especially with requirements on time and resources. And more long reads would always be better, of course. One last thing I want to talk about in just the last 2 minutes I have is a final project that we're going to be using Nanopore for. We don't have the data yet, but this is looking at long distance dispersal in plants. And so what this is, if you look at the phylogeny here on the left, you see that you have close related species that are in brown and in purple. You have the brown ones are in South America, and the closest relatives are in Africa. So this example of where you have things that are closely related in different continents is fairly common in plants. It's not super prevalent, but this is driven by infrequent, yet maybe more often than we usually think, events that have a large influence on gene flow and population structure. One paper suggested that if you were to move the population or to start a new population 34 kilometers away, you would need one event per year to establish that population. If you're looking at 415 events, or 415 kilometers, one event every 10 years, and so forth. So these are not very frequent events, but it does allow for colonization of vacant habitat and range expansion. So with Rob, we're looking at Larea, or the creosote bush, where you have relatives, or you have some species that are in North America, some species that are in South America, and they have diverged between 0.5 and 1.5 million years ago. And so a very short time in comparison to what it could be for such a long distance. We do know that so the king clone plants is about 10,000 years old, and then there's some fossil evidence a little bit older. And so Rob spent a lot of time working on this, and what we find, especially in California and the Southwest, we see hybrid zones where, say, in the Chihuahuan Desert, you see all diploids. The Sonoran, you're getting some tetraploids, and then in the Mojave, you have plants that are 6X polyploids. And you have mixed individuals or mixed populations in these hybrid zones. And then looking at the ecological niches of these, it's very clear that the different ploides do prefer different parts of that environment or that habitat. And so we are sequencing now currently one species from North America, one species of South America. There's about 6 species altogether in this genus. Doing this both with Illumina short reads, about 90x coverage. We're going to do 2 flow cells per species of the Nanopore. The genome's about 1 gigabase genome, so we should be able to get some good data from that. We will be doing the SRE XS, so it removes some of the really small reads, but not a bunch of them. We don't have a ton of material, especially for the South American species. And we'll try some of the different assemblers, spades, abyss, canu, to see how things go. What we hope to answer for this is what's the genomic consequences of divergence between continents, between North and South America? Can we determine the origin of the long-distance dispersal, where to begin? And what's the genomic control of ecological differentiation in the mixed ploidy populations? All right, so just to wrap up all the way here. So using the Nanopore with Illumina data has allowed for better determination of differential gene expression in the transcriptome assemblies when we're trying to find which parent the genes come from. The big genomes are computationally intensive, but even a subpar draft genome can help a lot, depending on what your question is. And then the more computational, there's many more computational options when it comes to small genome things. Things that I have found useful and helpful through this journey of Nanopore. Clean DNA for this is imperative. That may be a no-brainer, but if you have things that clog the pores, it doesn't work. Removing the small fragments, it does wonders for the output you're getting. So the short-read eliminator or the short-read eliminator XS has helped a lot too. As Fei mentioned, we've been doing nuclease flushes between different libraries of the same flow cell. That's been boosting our total output. The read correction can take a long time, especially with larger genomes. You can do that before or after assembly, depending on which way you want to go, but it does help. And then again, for the really large assemblies or large genome things, the normalizing the data has helped a lot with computational parts of it. So lastly, just acknowledge, so a lot of this work was done on my NSF postdoc fellowship, collaborators for these projects and others, and then it's been great having Fei around to talk to him about different things that him and Duncan were doing in the lab, and also Kevin Nixon for letting me use some computational resources over at the CUPAC server. And if there's any time, I'll be happy to answer any questions. Thank you. Yes.
So for the genome assemblies, you were showing separate results from Nanopore and hybrid. I think for the transcriptome, you only showed hybrid and Illumina. Did you also try to assemble those as a Nanopore?
I did try that, but we only had, so for each species, about 3 million reads, and I tried to assemble just with that, and I was not getting good assemblies at all. The number of contigs was like 1,000 contigs. So I don't know for sure what was going on there. something I need to look at. But I think even if we were just do just the Nanopore, I think because we had only 3,000 reads, I think we potentially be missing a lot of genes that we might be interested in. So I think if we want to do just the Nanopore, we want to bump up the number of flow cells. We just did the one, and we multiplex different things, or multiple samples together. So if that was the goal, especially for differential gene expression, you want to do a lot more flow cells for that.